From now on, we are going to talk about immunological memory. What is immunological memory? First of all, we'll show you the definition of this phenomenon. When our immune cells meet any germs for the first time, they learn how to recognize them. After this first encounter, some of these cells stay in the body as memory cells. By doing this, lymphocytes can respond quickly and effectively to antigens. This is how immunological memory works. Next, we'll show you what the biological roles of immunological memory are. One important role is to enable rapid immune responses upon reinfection. During the first encounter with germs, naive lymphocytes need several days to react. But surprisingly, during subsequent encounters with the same germs, memory lymphocytes can respond within just a few hours. Also, during subsequent encounters, memory lymphocytes can act more strongly to pathogens. This means memory lymphocytes can have more rapid and strong responses after the first time. Therefore, the immune system can eliminate pathogens more efficiently before the condition becomes severe. On top of that, memory cells have longer lifespans than other cells, so they save us for a long time. On the other hand, vaccines make use of this mechanism of immune memory by introducing antigens into the body and making memory cells. Immune responses in the actual infection will be improved. Next, we go to the mechanism at cell level. Two types of immune cells play important roles in immunological memory: memory B cells and memory T cells. Memory T cells can respond to smaller amounts of antigen than naive T cells, so they can proliferate and begin to act immediately when pathogens invade. There are two main types of memory T cells: memory CD8 plus T cells, which become killer T cells, and memory CD4 plus T cells, which become helper T cells. Killer T cells rapidly destroy infected cells, while helper T cells rapidly activate B cells and macrophages. In other words, they rapidly help other cells. Memory B cells are activated at a much lower level than naive B cells. After activation, they rapidly differentiate into plasma cells and produce large amounts of antibodies. Also, they can produce more effective antibodies by switching antibody classes. Besides, memory B cells and some plasma cells have really long spans, usually for many years. This property ensures the effectiveness of vaccination. Now let's move on to the gene level. Why are memory B and T cells so rapid? The answer is epigenetics. The chromatin structure of genes necessary for immune responses are loosened. Therefore, transcription begins immediately so that memory B and T cells are able to respond more quickly. Now let's focus on memory B cells. Memory B cells are able to produce stronger antibodies after the first infection, but why? Concerning this property, there are two key points during the first infection: mutation and class switch. First, let's talk about mutation. During the first infection, B cells introduce mutations into the antibody genes, changing the shapes of antibodies. This allows the immune system to select antibodies with higher affinity. Next, let's move on to class switch. During the first infection, B cells switch the class of antibodies from IgM to IgG. IgG is more effective than IgM. These steps are not necessary after the first infection. For these reasons, memory B cells are able to respond more quickly with stronger antibodies. This is the end of our presentation. Thank you for watching.